all. Uh, okay, so I should say this because I need to just one moment. Now my screen has become very big actually. Yeah. Okay. So let me begin by saying a very warm good afternoon to you all. I welcome you for this second session of this talk for this iHit Summit. Today we have three distinguished speakers, very eminent speakers. And the first speaker is Professor T.P. Singh. Professor T.P. Singh is currently served DST Distinguished Fellow at the All India Institute of Medical Sciences, AIIMS, New Delhi. And he has made outstanding contributions in the field of biophysics, particularly in the area of extracrystallography, structural biology, peptide design, and drug discovery. Presently, he is focusing on structural and functional studies of innate immunity proteins and their exploitation as protein antibiotics. So he has published more than 435 research articles, and importantly that he has contributed enormously by submitting 615 protein structures in the protein data bank. He is the fellow of World Academy of Sciences, Indian National Science Academy, Indian Academy of Sciences, National Academy of Sciences India, and Biotech Research Society India. Professor Singh also has received Professor G.N. Ramachandran Award of the Kerala State Council of Science, Technology and Environment. Is also the recipient of G.N. Ramachandran CSIR Gold Medal for Excellence in Biological Sciences and Technology. And Professor G.N. Ramachandran's 60th birthday commemoration INSA Medal. Shastra G.N. Ramachandran Award for Excellence in Science. I can go on, and I think so that we will listen to Professor T.P. Singh, and he's no what to say, like he's a well-known person, and you can actually look into his websites also. And so I will give the floor to Professor T.P. Singh. Professor T.P. Singh.
Thank you, Shriram. Thank you so much, and thank you, Dr. Raghav, asking me to be part of this health summit. It's very nice title of the conference. So I think in the present time when we talk whatever we do, but in the context of what we have faced in the recent times, and what we have faced in recent times, everybody knows this pandemic. So I'll come to this, but to begin with, I will make some general slides because of audience being very general in nature. Oh. Stop sharing. Oh, no, sorry, I made a mess of it. Can you see this? Okay. So just to give you that, when you talk about bioinformatics or whatever we do today.
I'm sorry to interrupt.
I cannot see your side, sir.
Can you see my screen?
No, sir. No, sir. You need to share the screen now.
Oh, where is the sharing gone?
Yes, sir.
Where is the share now?
So it should be in the bottom. I mean, the third option, the share screen.
Oh, it must be in green color.
Green color.
Yes, there is in the bottom, there are so many icons are there. So there is a screen sharing, which you can click it.
Oh, right. On my.
In your screen, right?
Just call this guy. Just 1 second, please. This slide primarily is, you can see for yourself that we have done great things in India. And if you see at number 5, molecular biophysics, basis of today's bioinformatics, Mr. G.N. Ramachandran's contributions have been the key, and why I brought this in line. This year is going to be his centenary year. He was born in 1922. So I think when we talk about anything on bioinformatics or health informatics or whatever related to this kind of thing, G.N. Ramachandran's contributions enormous. And so I will make a couple of slides before I come to my things. You see, as far as I see, his contributions have been the most outstanding from India, particularly in context of modern biology, and more specifically in bioinformatics. The kind of things he did have become the basis of all these kind of things. Just to give you an idea, to those who are not familiar, everybody will be familiar, but those who are not familiar with this, see his contributions in those years in 1950s and early 60s, phenomenal. His discovery of triple helical structure of collagen was an outstanding contribution. He gave he gave his 5' map, and also his group, these beta turns, how a protein chain folds, turns, reverses its path. All these things were given by him. And these things have become basis of our structure determination and validation of structures. Whatever we do with proteins, we need to use his contributions. So these things are very, very important that way. And this is his centenary year, so we are very grateful to him. So this is a tribute to G. N. Ramachandran. Since Raghav asked me to talk about something, India's contributions to DB, I would say that this initiation was made by G. N. Ramachandran in the 1950s by giving the structure of collagen and also giving the basis for validation of protein structures by this Ramachandran's 5' map. And also, I think, giving three very fundamental beta terms. The terms he characterized how when a linear chain of protein turns, folds back, what are the characteristics of these terms. So if you see from that point of view, at that time, it was a huge contribution. There were no computers, there were no idea of protein structures. So in that era, it was a fantastic contribution. You can see the main contribution he made in just about 17 years when he was in the University of Madras. So this is something very huge contribution from him and his center in Madras. Now when you talk about protein structure, this is very basic, but I think could have some relevance to who deal with not actual structure determinations, use the structures. When you want to do structures, need your protein samples converted into crystalline form. Proteins you can prepare whichever way, whether you isolate from natural source or you clone and express them. Need to convert them into crystalline state because individual protein molecules, when they are put in X-ray beam, you won't see much, but when you put large number of millions of molecules in an ordered arrangement, their contributions are added and you observe some kind of diffraction pattern. When this diffraction pattern is analyzed, you see a wonderful structure of proteins, globular proteins, folded proteins. And these proteins are folded in a manner that they all have some kind of functional site known as the binding site. Binding site in all these proteins is very uniquely made. So this is very important for all of us, those who want to understand the structure-function relationship, or those who want to understand the functions of proteins. Knowing their structures, knowing the details and the stereochemistry of the binding sites are very essential to explain the function of these proteins and also modify their functions and regulate their functions, to block their functions or to enhance their functions. So these structural details are fundamental, and they are all the structures of these proteins part of a public database, protein database. Just to give you an idea, when this began, the first protein structure came late, maybe 1959 or 60. And then some more structures came in 1960s. A few more came in 1970s. So this database was created sometimes in early 70s. You can see that the number of protein structures in database grew rapidly. It is growing much more rapidly now. So lots of locations. So I think take a clue from Raghav is this, that you don't have to only consume what others produce. You must contribute to things which others can consume as well. So if you see this slide, this is from the database, just to give you an idea that most parts of the world contribute a different magnitude to this protein data bank. So to have an idea that there are locations where you get more structures deposited, but this is spread all over the world. And India also makes substantial contribution to this database, although there are some white regions which should become dark. That is needed. So we should contribute as hugely as possible. And as we have enough human resource, and other resources are enough, must contribute to this. Although most of the times, because there is a rate limiting step, we had to convert it into crystalline state, and many times, many scientists use patience and then they move on to this computational biology, not exactly computational biology, they start looking at talking in molecular modeling. I think we need to have patience and we need to keep trying structure determinations, even though they take time, even though I think they may not publish that extensively, but it's important that we carry out experimental work as much as possible. So this is the status of India's contribution to this. If you see in the map, I think it's quite significant. The four major regions is East USA, Western Europe, India, and Japan and China, and to some extent, the continent, Australia and New Zealand. Now, just to give you an idea, this I think is not relevant anymore in the present times. It was only relevant until some years back. The individual labs were considered to be important contributors. Now, contribution is quite widely spread. There are a lot of centers, not necessarily individual investigators, where structures are being deposited. Historically, a decade back, these things made sense. So some labs which made contributions, just to give you an idea. So it is in the historical context, not the present times. Present times, a lot of locations have emerged, which might not belong to an individual investigator. They contribute quite a lot to the structures. Now, if you see, this is very important as well. See, those who submit to the protein data bank and those who exploit protein data bank, you can see from these contributions, 3 continents use it very extensively. This is well known, but at the same time, you see you expected more from South America. And of course, Australia and New Zealand, significant contribution. Africa, as usual, we know that happens in that way. Now, if you see the country-wise, some kind of, it's all from the PDB side. So if you look at this, people like Raghav must be very happy that this country is only number 2 in terms of using this database. So this is very fine. I think this shows that we are very much involved, but maybe more serious output has to come out, and maybe we should produce more drugs because we are using it quite extensively. This was in some years back. Here was this 1230, By now, these numbers have grown. Oh, this has become about 10% now. This is how you see the scenario for PGB. Now, coming back to a little bit, we have everybody in the world, majority of scientific community, the world has been addressing this issue of pandemic. And this has been a very massive pandemic. You can see that so widespread, changing all the time and becoming very difficult. So lots of investigators got into this. Everybody who was working in biological sciences, some way or the other, directed their efforts towards addressing issues about this pandemic. There have been millions of scientists who have been involved, and massive number of new publications and results have been made. And of course, we are lucky at this moment. Vaccines have come, but I think eventually the solution would be that we understand the disease, we understand because the virus doesn't have many proteins. Many of them are very critical proteins. We understand their structures and functions, making drugs against them the right solution. If you have potent drug molecules, there's no need of vaccine to control the virus quickly, and that is the need of the hour. Do not have yet anything. Lots of molecules are there, but they are not really serious at this moment. Eventually, with more knowledge and contribution in this field, we might get into making potent molecules. So just since it's a virus, not many proteins, that many of them are critical for its progress, its survival. Some of these proteins are listed here. So these proteins can be good targets. So there is a massive contribution from those people who use this bioinformatics tools. Definitely some advancement has been made. I think many structures are also there. So it's a matter of some time, maybe a short time, that we will have some kind of potent molecules in the future. But if you see, how do we in general fight infections? It's just for the general audience. In principle, our innate immune system should be able to protect us from all the viral and bacterial infections. And we assume that this innate immune system is a part of our germline. We don't need to do anything. This is there, and it should automatically stop us. It is not able to stop us. Particularly in this case, you've seen so many people suffered and so many casualties occurred. So there is some issue with the innate immune system. So it would be important to understand what is lacking with the innate immune system that we are not protected by that. And if you understand this, is there a way to make it stronger so that it could fight infections better? And we don't have to go to the next step where start looking at antibodies and start looking for vaccines. So if innate immune system could also be looked at in some way, that why it has not been able to do that, and whether varies from people to people. As you notice that many parts of the world, this pandemic has been much more severe than the other parts of the world. So I think there's a need to examine what is going on with our innate immune system. So that's one thing, and that's the focus of my presentation today. But we all must appreciate that very quickly many vaccines have emerged and more are coming. But lots of people take one dose, second dose, booster dose. This is an unending process. We have to think about other means, although for the time being, it's a situation, we should appreciate that we have some kind of relief. And also virus, there are lots of mutations which can be bad, but there are lots of mutations which sort of make virus weaker. So this process goes on. But to just give you an idea that We were very lucky many years before when antibiotics emerged on the scene. See, when the crisis occurs or when things are not there, something or the other occurs. Sometimes things will happen by chance. Sometimes your knowledge will drive to take you to the solutions. So it's good to look at how people in the ancient times fought these infections, how antibiotics emerged, just some historical perspective, just to give you an idea of how civilizations and how industrial advancements made differences. As of today, we have a lot more advantages, but we have seen that in spite of that, we have difficulties in fighting this pandemic very quickly. The reason is that if you see the last wave, synthetic antibacterials, something or the other will happen, like now vaccines have happened because the antibiotics were discovered by some sort of not very scientific approach. So this synthetic antibacterial did not make serious progress, as you would see from the next slide. Before we go to that, why synthetic antibacterial didn't emerge, this is just some perspective that historically, this is very important that when you have civilized minds, when the minds are trained to think, and they think in terms of positive aspects. You can think of solutions even though you don't have methods, you don't have tools. So this slide is only to highlight that this part of the region is civilized. People were thinking how to make lives better. So when these sort of infections occur, they thought of a lot of materials. They used many things, and by way of observations, they found some of these materials were very helpful in controlling infections or fighting infections. So in other words, if our minds are trained, we become civilized, we are not crazy doing nonsense things, we can produce, even if we don't have that kind of great support, and that's how they generated so many materials. So this was just to highlight that civilization, positive thinking minds can always find solutions, even though they did not have that sort of support what you have today. This is just to highlight that India's civilization is one of the oldest civilizations, contributed a lot of knowledge in many areas, including drugs, particularly. You see these molds were used primarily in this part of the world. And if you see, when some other part of the world gets industrialized, they take clue from these materials. They start looking at as to what these materials contain. If you see in the next slide, you will see the geography changed, and they started using these molds, the same materials which were used by previous locations. Begin with, they just recommended it straight away. And slowly, when the tools emerged, industrialization occurred, they started sort of thinking further and started isolating things. If you see this time period, a lot of things happened, some incidentally, and some using methods. Finally, first antibiotics were identified. You read these things, it's a very interesting development of this kind of things. You know, these stories are very great, and that's how the science grows. So this grew in a certain way. I think in a parallel way, things would go even now, and we will find solutions to this sort of difficult pandemics as well. So this is what I was saying, that because these antibiotics came, antibiotics came, and then of course, when one thing happens, then many things happen. So many antibiotics, many general and specific bacterial viral infections, they emerged. So scientific community was focusing on that side. Industries were more concentrating on that side. So synthetic materials were very few. At that time, there was not much of bioinformatics. There was not much of molecular modeling and docking approaches. So you see, people made very few. Sulfonamides came in 32. Trimestine cyclic peptides came in 39. In 1962, some quinolones and its derivatives were made. So very little progress, primarily because all potent antibiotics were there. Now, I don't have to talk about details of this. I think there will be some people talking about this, but primarily, these are the four methods the investigators have used. Some people still continue to examine natural products from plants, medicinal plants, and so on. There's so much emphasis by the government, also the government of the day, that we should think about our ancient approaches also, but to be very polite in this case, I would say that I think this is a matter of past. You cannot expect plants to have solutions or natural substantial solutions for very specific problems. But this thing anyway goes on. And lots of people in the chemistry labs and industry keep on playing with ligands and molecules, adding moieties, deleting moieties, making combinatorial approaches. And they make a lot of molecules that way and try them, and this process goes on. And of course, finally, the most traditional approach is that you identify the target, have information about the binding site, knowledge about stereochemistry. It's very, very much possible these days. The structure is known. It's very, very easy to have an idea. And based on this, it's very easy to suggest a molecular structure which can be synthesized by chemists, and then you can try this. This approach can produce molecules, more potent molecules in a time frame very quickly. Since many of these things are still in a phase that we are not making great progress in terms of producing really good drugs. Some are coming out, so lots of people are just blindly making compounds after compounds, use synthetic libraries and using some kind of throughput screening to look for some luck that something might be binding well and might be a potent drug. And the limit, I think, is there are lots of people. In the present time, they started repurposing. These are very blunt approaches. They are not. Sometimes something very approximate may happen, but the real molecules cannot come out by these methods. Need to become more scientific. And to be more scientific, I think this approach has to become, whether you use computational biology or your experimental science, eventually we will have to come to this, that we follow targets, learn about targets, and we need to have knowledge, need to understand what these targets, what kind of binding sites are. And once you look at them, they will give you a clear idea as to what you need to design that, and then the chemistry becomes very important. I think chemistry has been lacking in many instances. Hopefully will improve better, but if you follow this path iteratively, I think it will be the real way to achieve that. But I'm going to give you some different ideas today. As I said, that we haven't done much with innate immune system. So many proteins which contribute or which are involved in fighting infection, which interact with these invading pathogens. What is lacking in them? Can we understand how they sort of interact and why they are not able to prevent what they should be preventing? So a group of proteins we have been looking at, And we think there's an interesting way to address these issues. Now, if you see these innate immune actions of some of these proteins, and if you see that these proteins are secretory proteins, they are present in all these secretions, primarily all secretory glands, they are synthesized. So they are secreted, and they are at all those sites which are the entry points for these pathogens, virus and bacteria. And they try to fight infections. For example, some of the proteins which are secreted from these fluids, you see, particular class of proteins binds to cell wall members. They recover. Innate immune action is generally a very general action in the sense that they have to have one protein can interact in certain way and will contribute preventing, protecting you. There would be other protein will look at different way. The other proteins, so a group of proteins, if you see that this particular class of proteins to try to recognize cell wall molecules of bacteria, you have another protein which primarily sequesters iron with very high affinity, depriving bacteria and virus of iron. Even lysozyme hydrolyzes cell wall PGA. And this mammalian heme peroxidase, lactoperoxidase, it is an enzyme and it has a very complex mechanism, but produces with the help of hydrogen peroxide using substrate like thiocyanate, highly antiviral antibacterial agent hypothiocyane. This reaction is known. So this is an enzyme which does it. It's a part of the secretions from all those sites, and is highly expressed when infections occur. So this enzyme is a member of 4 mammalian heme peroxidases. See, now this particular one is secreted at all these sites. Other heme peroxidases are at different sites, but to our interest, this is important in the present context because need something which is present in your nose, need something which is present in your mouth, which is present in other secretory sites. And since this is an enzyme, now we can ask questions, what is there? Why, if this expression of this enzyme is very high during infections, why are not protected by this? And the issue can be addressed in this way. This is just a little bit, tell you a little bit more, but how does it work? How does this enzyme work? See, it's hydrogen peroxide. Now, hydrogen peroxide is produced by this kind of reaction in the system, that glucose is converted by this enzyme into gluconic acid and H2O2. So H2O2 production is not an issue. This enzyme is glucose oxidase is expressed in high concentration. Glucose is abundance in human body, in animal body. Now, H2O2 production is generally not an issue. In fact, H2O2 production very often is higher than required. So there are enzymes to take care of excess H2O2. So H2O2 is not a problem. If you see the output of this particular enzyme, which should be protecting us. Now it uses a substrate. Once H2O2 interacts with this enzyme, an intermediate is formed. This intermediate becomes the next step, which converts substrate into antibacterial product. Now, is this substrate concentration is a problem? H2O2 is not a problem. There are 2 substrates. First step H2O2. Next step, real substrate converted into highly reactive, highly antibacterial, antiviral agent. So that is the issue. Is this a problem? And if this is a problem, we address. So it is something like this. If you see the scheme of things, the particular step where H2O2 is produced, there is no problem at all. If you have a problem, you can easily supply glucose and this production can be enhanced. The expression of this enzyme is not a problem. And there are other steps also, I'm not giving details here, by which way S2O2 is produced. So availability of S2O2 is not an issue. Now substrate thiocyanate, its concentration varies over a range and it's very difficult to supplement this. It's also used by some kind of enzymatic steps, converting cyanide and other stuff into SCN. So that step, we still don't know how to enhance production of SCN. Instead, we think of some other substrates here. And if they could be also converted by this enzyme into this antibacterial product, we can fight these pathogens, these infections very easily. The issue here is that this member of the innate immune system is an enzyme, and this enzyme uses substrate. So could this substrate be supplied? Most of the time we talk about designing inhibitors and that sort of agent, but here we need to think of substrate, how to design a substrate. But we need to understand that this particular reaction is complex in the sense that first step is with S2O2. It's a very sensitive step again. You know, when you talk about enzymatic reaction, it's not just mixing. The S2O2 has to be placed in a very appropriate way, the binding site, to produce the first intermediate. And then SCN has to align. Because they are very small substances, they have to be aligned by interactions at the binding site. So you will see that how this is important. In other words, you need to look for new substrates which can be supplied through dietary means, or which can be supplied by oral means, or which can be given through intravenous routes. So this is the question here. And this is how, if you have found out more substrates, we are strengthening your innate immune action by this particular protein, member of the innate immune system. So that is the question, whether we can very quickly exploit this sort of situation to fight pandemics as we face this time. Now, how to look for substrates which are very appropriate. See, it is not that you may design a compound. You may design a compound, but it may bind to this, but it may not act as a substrate. So you have to very critically align in a way that design becomes very complicated, how to think about such a design. But a lot of things happen, and some observations were made by many people. Reports were made in journals and in newspapers that in the initial phase, people in Japan were less infected. It's a country which is a fairly crowded country, but infections caused less. And one of the factors was investigated. It was observed that the iodide content in the population in Japan was relatively higher than the rest of the world, particularly from the Western world. The iodide content, if you see, is very low. Although recommended value is very high, but generally people consume much less. I mean, India and this country, this part, you have something of this order, whereas in USA is much lower. Japan is relatively high because their diet, their seafood, meats they eat, and it's fairly high. So that was considered to be one of the reasons this argument was made. So we thought that could iodide be substrate of choice? Could this be one of the possibilities that the people are protected, that iodide is used by lactoperoxidase to convert into hypoiodide, which is a highly reactive product. So the best way is that, just to give you an evidence that we had been very active on this, on the structural studies on this protein. In recent times, we have enhanced our activity much more. So we did many more publications in recent times, but this became an important. Wanted to see that whether this could be one of the ways to fight infections. This particular enzyme that we can have these substrates and produce this real antiviral agent. So what normally one would do, as most bioinformatics people do, that they try to look at the structures of these proteins, since we have the structure of this protein in great details from a number of sources. So there's quite a bit of knowledge about the binding site. So in the zoomed picture, you see, this is a cartoon diagram, but just to give you an idea that substrate binding cleft is fairly deep and is well understood now. So we have an idea about this. To understand more and more, you look at this, and more you look at this, get ideas as to what you can try. So we thought that we should try something with this iodide. We tried with many anions and many anions there, that they can fit into this halides, other halides, iodide and other halides. So the many structures were examined, but and some small organic molecules also. What I want to say is that we examined many halides and some small organic molecules, but in a positioning of these halides or other molecules, the substrate in the binding site has to be such a way that it has to be, when the enzyme is converted into intermediate, the first step, the hydrogen peroxide, the binding site has to align in a manner that the critical reaction of what proton transfer mechanism this enzyme does happens. So many halides doesn't happen because the alignment is not appropriate. So this, I don't have time to give details about how the neighboring residues contribute to this alignment and how they promote the proton relay mechanism. But this is very critical. This is what I want to say. Bromide doesn't get converted by this protein, but halide, this iodide gets converted. Chloride doesn't get converted. Fluoride doesn't get converted. It's a very specific alignment. So these studies have revealed, when we did many complexes, that iodide is the one which is converted by this particular protein into hypoiodide, and that too is based on we observe hypoiodide in the binding site, and because the evidence, you can only say that you have observed this. So I'll come back to this, but the idea is that this reaction, if you see, the first step produces here, this is different enzyme produces H2O2. H2O2 and this protein produces this compound 1, the intermediate of LPO, lactoperoxidase. And this is the one where your substrate binds and produces this highly reactive antiviral agent. Now, to understand this, you need to have look at little bit more details. So first you do this protein structure, the unbound state, free state. So very quickly, you have to do this by observing experimentally. And the way you observe experimentally in this methodology is that you observe electron densities, and the electron densities, you define these things. So unbound protein at this binding site, site is known as distal heme cavity. You have only water molecules. So you understand that this histidine is a critical residue. I'm not giving details, but to the general audience, this is how this typical site where reaction occurs. Now next, we try to see whether H2O2 can bind to this. See, you might be knowing that we talk about enzyme substrate reactions. You cannot observe substrate bound to enzymes where they get converted, but this enzyme has two substrates. So if you do not, this is another interesting observation. If you react to your enzyme with iodide and add H2O2 afterwards, H2O2 can bind to this, but cannot produce intermediate because it doesn't attain the orientation. if I do not have this iodide, S2O2 quickly gets converted into compound 1. You cannot observe S2O2 here. But if you can place some molecule here, including iodide, before you add S2O2, S2O2 doesn't align. In other words, S2O2 has to bind to this enzyme first, and then substrate iodide has to go, then you produce a product. But in this case, we
Professor Singh, can I just interrupt and ask a
yeah,
question?
please do interrupt. Yeah.
You are saying that H2O2 cannot the H2O2 cannot bind to your enzyme unless and until that it binds the iodide binds to your H2O2 and then it actually binds to that enzyme.
Yeah.
So have you looked into the case of any cooperativity or allostery in this particular mechanisms actually?
Yeah. No, what I was trying to say is that H2O2 binds to this straight away. When you mix this enzyme with H2O2, get a reaction product invariably, there is no problem. But reaction product you get with H2O2 has a opportunity to align appropriately with histidine. Here I have not given other arginine here, just to align very appropriately between heme moiety and the histidine. But if you have placed a substrate already here, other substrate or inhibitor here, H2O2 goes and binds, but doesn't align appropriately. That's why reaction doesn't occur. So we observe that way.
So it's just a sort of a competitive reactions that is happening, or what is that actually?
Yeah, a competitive reaction, but there is no iodide doesn't go in here because of its size. Right? Only H2O2 goes here. So this, I will come to this because there are more results to show this.
Yeah.
So this is just to show you that where H2O2 binds in this, orientation has to be very critical, but approximate orientation you see, and that you observe very nicely with the electron density. Here we observe this, and the iodide substrate binds here. So this observation gives you an idea where substrate binds, with the substrate number 2 iodide binds, the substrate number 1 H2O2 binds. This structure reveals this. Now, there are no well-known substrate is thiocyanate. So we observe how thiocyanate binds to you. When you make the complex with this enzyme without H2O2, you can observe thiocyanate very nicely. And you have an idea that thiocyanate binds here. And this site is this water W1, the H2O2 comes and binds, converts into this. But the important point is that enzyme and H2O2 have to get together first, and then your substrate has to enter. Then you get a product. If you give your substrate HCN or iodide before H2O2, H2O2 will go and bind here, will replace water molecule, but will not produce a product because it doesn't gain that required orientation or very sensitive proton delivery required in this reaction. So anyway, that details is a different apart, but just to gain ideas where these substrates are binding. Now, always when the product is made and if it's not being consumed, so you make this reaction with H2O2 and SCN, you get a product, but if your product is not utilized, the product remains around and it goes and binds and inhibits the reaction of the enzyme. It's a usual thing in most enzymatic reactions. The substrate product is not utilized. It goes and binds and inhibits the action of enzyme. So we observe where SCN binds, when particularly it is not utilized. Enzyme action is not required because it's not being utilized. Now then we did exclusively similarly with iodide. We saw that with SCN, and we saw iodide ions. Iodide ion can't go and replace W1, but it locates here. 3 iodide ions binding here like this. This position is exclusively for H2O2, the W1 is. So you have an idea that iodide is binding, and its position here is very appropriate. If H2O2 is already bound, iodide position is very appropriate, which we didn't observe with bromide, chloride, and other fluoride. So it's then the issue was this: you see the binding of iodide at the binding site, and you say that you could see the product, but how do you observe the product? Until you observe hypoiodide in the reaction product, the structure you demonstrate, cannot claim that this is converted into product. I am not giving many slides, but we very systematically examined, and we found that hypoiodide ions were observed here. So what you should do that when you create your reaction with H2O2 and enzyme, and then an iodide, and if the product is not utilized, which is not utilized in the catalytic, the crystallization condition, then you observe hypoiodide ions. And the observation of these hypoiodide ions, the substrate binding site, shows that iodide is converted into hypoiodide. The idea was to prove that iodide is converted by this enzyme into hypoiodide. And we haven't observed this with chloride, bromide, and chloride. So exclusive substrate for this iodide acts as a, I think my time is running out, just quickly finish. So we found out that iodide can be a very appropriate step to produce this antiviral agent by this reaction. Then we also wanted to look at more substrates. There were some observations, some reports about nitric oxide. Then we placed nitric oxide and observed here. And when we made the reaction in a similar way, you observe nitrite bound here. So nitric oxide is converted into nitrite, and nitrite is also antiviral. In other words, we identified that iodide is converted into hypoiodide, nitric oxide is converted into nitrite, both are antiviral. So it's a very selective, very critical reaction, which shows this. So then we had done more reactions, more complex, but I think I don't want to take more time because time is exceeding. Now, what you can conclude here, what was known biochemically, that SCN was converted into hypothiocyanate. So we identified new substrate, iodide and nitric oxide, which can also be converted by this enzyme into highly antiviral agents. We are also looking on some small organic molecules, which could also bind to this and are converted into highly antiviral agents. So, but as of now, you already have iodide and nitric oxide, which could be exploited when you have a situation like this pandemic. These two substrates could be supplied, then you will have control on and the spread of this viral infection. And these are the sites where this enzyme is expressed in high concentrations. Lack of substrate perhaps makes it, is a real lacuna there. And by giving these two substrates, we may control this situation. And now this reaction mechanism, we need to find out more substrates, maybe more organic molecules. But these two are new substrates we can add to this. And these can be supplied through dietary means, through nasal spray, and the more ways so you can supplement the deficiency of thiocyanate by these two substrates. And this is recommended as of now. Want to remain protected, have more iodide contents in your diet, more iodide content by other means, and more nitric oxide content. You will have better protection against virus. So this is a real finding of these studies. So this is indirectly sort of addressing the problem, addressing through innate immune system. So this is the first example where we can claim that innate immune system is not just germline step that which may have limitations of, I mean, there are many other members where you cannot do much, but this being an enzyme, using a substrate, using an internally synthesized substrate, you can supply it externally and to strengthen this and enhance the production of antiviral agents in your body and you protect yourself. I think it would be better that is time running out, but you can have some couple of comments and questions so that you can have some interaction. Thank you so much.
Thank you, Professor Singh. So if there are any questions, the floor is open for discussion from the audience.
Close the screen or?
No, sir, there are no questions from the audience, I guess. I mean, they have not typed any questions Q&A.
Okay.
Nice
So,
try.
Professor, I do have a couple of questions here, right?
Yes.
So one of the things that which you are mentioning is about the iodide, but the other halogens are not playing any role at all.
Right.
So is it because of the size of your iodide, because that is much bigger than your chloride or fluorine, which is...
You know, it has to reach that crystalline cavity, which is deep inside in this protein. And
Right.
the only way it can be stabilized by, there are some positive charge interactions, arginine is there, histidine is there on the other side, there's a glutamic acid on the other. The glutamic acid is far away, but it's a repelling component. And there are some sort of van der Waals contacts involving this. So it's a positioning of this particular halide ion is very critical. That's why iodide is being converted and not chloride. Size matters here.
Okay, so it is basically about the non-covalent interactions that might be playing. Okay, so but how do we then explain it in the case of nitric oxide? Is again basically the oxygen being highly electronegative, so there is a possibility that like the electronegativity of the oxygen plays an important role, like I minus, which is also an electrophile.
Negative charge is needed there on this, but again, size-wise also, this aligns very appropriately like iodide. So that's how we could see product. Actually, any negatively charged stuff, many compounds can be converted by this enzyme into, can be oxidized, essentially oxidizing them. So, but they have to be placed appropriately at that site. That's the most critical thing.
Okay.
So, this is what we need to find out, which can be oxidized by this enzyme. And the best way is to place them in the binding site and see whether they are placed appropriately. Actually, mechanism is very interesting, but it's slightly complex and can't have time.
I was about to ask you about the mechanism, since you have just pointed out. This is a simple mechanism and enzyme kinetics of reaction, or is it a multi-step reaction?
It's a multi-step complex reaction. It's and actually, I didn't say this, but there are many, many ways that this enzyme's reaction can be hampered. So many ways. So it's a very, very sensitive and complex reaction. But if you have an appropriate substrate, then the chances are that you will have no problem going on in your in vivo conditions.
Okay. Of course, just a last question is that like whatever you have projected here is a speculative mechanism. I mean, in terms of basically the substrate and product, that is fine, which you are very sure. And you have also shown it through the structure, actually, the electron density structures and everything. Yep. But what I'm asking basically is that like whatever the diagram you have drawn here in this particular slide, namely the iodate and nitric oxide going into hypoiodate and nitrite, it is you've written like a simple one-step reaction, but it is a multi-step. It's just a speculative mechanism.
Yeah, there's several further steps in this. I not mentioned them here. You see, this is not speculative in the sense that we have actually observed them. We have observed the product in the binding site, which is an evidence that iodide is converted into hypoiodite and nitric oxide is converted into nitrite. Right. So these are observations. So ambiguity is not there, but from this second step reaction, there's multiple steps.
Right.
It goes, because in, I think in a popular seminar, perhaps it's not
Okay. No, what I meant was that I asked about the intermediate step, which are speculative. I'm not asking about the substrate or the end product, actually.
We have also observed some structures of intermediates, which I have not shown here.
Oh, okay.
We have done more than I think around 90 structures of this particular protein from different sources, and many of these situations have of them intermediates. So you say the most important point what we were looking for was that can we observe the product of some of these substances? And in these two cases, we observed the products experimentally. And that is an evidence that this is converted. These are converted into product, which is antiviral. And I think these studies go on and on. There are more interesting examples. Maybe we'll have next occasion to talk more details on these things.
So are there any other questions?
Where is Raghav?
No, sir. There is no question.
Okay.
Where is
So,
Raghav?
yeah, please, Professor Singh.
I was looking for Dr. Raghav, is gone.
No, he is very much there.
Yeah, I am here actually.
What is your comment? You are the crusader.
No comment.
No comment is a good comment, I would say. Okay,
Thank you very much, sir.
thank you. Thank you, Shri Ram, and thank you, Raghav. And thank you, everybody else.
Thanks. Thank you, sir.
Thank you very much for